All right, let's dive right in. We're going to tackle a really wild idea today, one that's kind of fascinating but also a little unsettling. Can the trauma your ancestors went through actually be written into your own biology?
I mean, seriously, think about that for a second. Can the stress your great-grandmother felt as a baby change how your body responds to danger today? It sounds like something straight out of a sci-fi movie, right? Well, that's exactly the question we're going to unpack. So our jumping-off point is this really incredible study from 2023, published in Science Advances. These researchers were digging into this exact question. Just how deep do the echoes of early life stress really go? And what they found? Well, it's pretty mind-blowing. They found real evidence for what they started calling a biological memory, a physical trace of hardship that can get passed down, not through stories or lessons, but through our actual self.
Okay, so how on earth do you even begin to test an idea like that? Well, it all starts with a pretty clever and maybe a little bit chaotic experiment involving some lab mice.
The technique they used is called repeated cross-fostering, or RCF for short. Basically, it's a way to simulate the stress of a really unstable early environment. What happens when the bond between a baby and its caregiver is constantly disrupted. To do this, instead of being raised by their own mom, these newborn pups were constantly shuffled between different mothers.
So here's the play-by-play. On day 1, right after they're born, a litter gets put into this RCF group. Then, for the next 3 days, which are super critical days in their development, the entire litter gets moved to a brand new adoptive mother every 24 hours. Yeah, you heard that right. That means they have 4 different moms in just 4 days. After that crazy period, they finally get to stay with their last adoptive mom until they're weaned. Now, this is the really important part to remember. This first generation, let's call them the F0 generation, they're the only ones who actually lived through this chaotic upbringing.
All right, now for the results. And honestly, this is where things get really, really wild. What the researchers found was that this early stress didn't just disappear. Nope, it created this lasting legacy of being hypersensitive to anything harmful.
First up, they tested how the mice reacted to a feeling of suffocation. They did this by pumping some extra carbon dioxide into the air. And just look at this chart. The mice from that RCF lineage had a way stronger respiratory response. It was almost like a panic attack compared to the control group. Their bodies were just totally overreacting to the threat.
And it wasn't just their breathing. They also checked for pain sensitivity by using a little heat source on their paws. And what do you know? Same story. The RCF lineage mice pulled their paws away way faster. It's like their entire danger alarm system was permanently cranked up to 11.
But here it is. This is the part that just blew my mind. This hypersensitivity wasn't only found in the original mice who went through all that stress, it was also in their kids, the F1 generation, and in their grandkids, the F2 generation. And here's the absolute kicker. Both of those generations, the kids and the grandkids, were raised totally normally by their own biological mothers. They never experienced that stress themselves, not once.
So what does that even mean? It means this isn't something they learned. It's not like a stressed-out mom was teaching her babies to be anxious. No, it seems like the memory of that trauma was somehow passed down biologically, like a ghost in their genes, an echo of something they never went through. Which, of course, brings us to the next massive question. If this is a biological memory, how is it being stored? What's the actual molecular switch that got flipped by that stress and then handed down from one generation to the next? Well, the researchers started looking closely at these proteins called acid-sensing ion channels, or ASICs. The easiest way to think about them is like tiny little sensors on the surface of your neurons. Their main job is to detect changes in your body's pH level, its acidity. This is exactly how your brain realizes there's a buildup of CO2, and it's also a big part of how you feel certain kinds of pain. And you guessed it, when they looked at the brains of those RCF lineage mice across all 3 generations, they found something incredible. The genes that create these ASIC sensors were working overtime. But not everywhere in the brain. It was really specific. The changes were concentrated in the medulla oblongata and the periaqueductal gray. And what do those parts of the brain do? They're the command centers for breathing and pain. So literally, the danger-sensing parts of the brain were being flooded with extra danger sensors. The researchers themselves put it perfectly in their paper. They said that this early life stress created a biological memory that you can trace right down to a molecular sensor, these ASICs. And this one molecular change reshaped how these animals dealt with threats for 3 full generations. So if this whole thing is a physical molecular switch, well, that leads to a really exciting idea, right? Could you maybe turn that switch off? Could you actually erase this inherited memory?
And the answer, amazingly, was yes. The scientists used a drug called amiloride, which is known to block these ASIC channels. They gave a single dose of it, inhaled, to the hypersensitive mice from all 3 generations. And just like that, their panicked breathing, their heightened pain response, it all went back to normal. It was literally like they flipped the switch back to the off position, completely silencing the echo of that ancestral stress.
Okay, so far this sounds like a pretty neat and tidy story, doesn't it? Stress flips a switch, the switch gets passed on, a drug flips it back. But, science is rarely that simple. It turns out that first study made a pretty big assumption about what was happening in the nest. And another study, well, it really complicates the picture.
See, the Battaglia paper sort of assumed that the stress was just about the separation itself. They reported that the adoptive moms were providing perfectly normal care. But a different study from 2020 by a team led by Finkler, they dug a little deeper. They asked a more fundamental question. What did those pups actually experience during that time? And this is where things get really interesting, because you see this scientific debate playing out. On one side, you've got that 2023 Battaglia study in mice, saying maternal care was basically normal. But on the other side, you have the 2020 Finkler study. They used a really similar RCF method, but in rats, and they found something totally different. They saw that RCF actually reduced how much care the mothers gave and made them less likely to retrieve their pups. They said it created an unstable environment that just completely disrupted the baby's ability to form a secure attachment.
So where does that leave us? Do we have a flat-out contradiction here that just tanks the whole idea? Or is there maybe a bigger picture that can bring both of these findings together?
Yeah. What if these findings aren't actually contradictory at all? What if they're really just 2 sides of the same coin, each one showing us a different but equally important piece of the puzzle? Let's try and connect the dots here. That unstable environment Finkler's group talked about, that could very well be the reason for the biological changes that Battaglia's group found. It could be that this specific disruption of attachment is the real trigger, the stressor that tells the brain, hey, ramp up those danger sensors. And this is so important. This isn't just a story about mice and rats. This whole mechanism has stunning parallels to what we see in humans. We know that childhood parental separation in people is linked to that same over-the-top reaction to CO2 and a higher risk for panic disorder in adults. And guess what? Human ASIC genes are also directly tied to panic disorder and how we feel pain.
The Finkler study really hammers this point home. They said the real problem with this RCF setup isn't just about swapping moms. It's that the constant swapping creates an unstable world where that fundamental process of learning to attach to a caregiver gets broken. And that, it seems, is the original wound that echoes down through the generations.
And all of this leaves us with a really profound, maybe even a little bit spooky question to think about. How much of our own biology, our anxieties, our reactions to stress is an echo of a past we never even lived? The science seems to be telling us that our bodies remember far more than our minds ever could.